Hello, and welcome to the 15th episode of the Doc Talk with Liz podcast. Today, our guest is Adam Beitz. Adam Beitz is a graduate student studying chemical engineering in the Galloway Lab at MIT. His current research focuses on building genetic controllers and characterizing the role of P53 in cellular reprogramming. Before coming to MIT, Adam obtained his Bachelor of Science in Chemical Engineering from Clemson University. In this episode, we discuss his journey into engineering, his advice for those beginning their careers, and what he enjoys doing in his free time. Adam, thanks for joining today. It's nice to see you again after some time of a long break over winter.
Absolutely, yeah.
So I guess I just wanted to start off by asking you about your journey into science, or more so engineering. I know you went to Clemson for undergrad and everything, and your parents were both school teachers, your dad with psych and your mom with English. But I kind of was wondering like where your inspiration for science came from. Just seeing as like a lot of people that are in science, I find do have parents that might have been in it previously, but that's not exactly your situation.
Yeah, so I think my story is somewhat typical of a lot of people who get into math and science, or who get into engineering really. Growing up, I was really good at math and science in school. And so people are like, hey, like you should do engineering. And I'm like, cool, like what is that? You know, like what does that mean? I took chemistry as a sophomore, which was like at my school happened to be like a year ahead based on the curriculum that we were doing. And I took like biology and chemistry at the same time. And I did really well in chemistry with a teacher who was known to be like super difficult. I got along really well with him. We started talking and he suggested chemical engineering. He's like, you're really good at chemistry. You're really good at it. You're thinking engineering, they have a chemical engineering thing. And I was like, oh yeah, that sounds cool. At the same time, I was like taking biology, and I think I was actually like drawn more to biology at the time. But I knew like chemistry also just like made a lot of sense to me. So I kind of rolled with that idea. When I was applying to undergrads, I kind of had in my mind, oh, like chemical engineering, like I hear that's a great field to be in. There's lots of jobs right out of undergrad. Like let's go with that. We'll start there. We'll see what happens. And so I ended up going to Clemson. It ended up being kind of honestly the most financially responsible decision I could have made. And that was really nice. It was nice to stay near home.
What were your other options, like your other top, or if you were to kind of go back, like your other top choices?
So the other places I applied to and were accepted to were University of Michigan and NC State University, North Carolina State.
Okay.
And so 2 out-of-state schools, and good opportunities, both really good schools. It just was 10 minutes down the road. South Carolina has some nice in-state scholarships. And so I did what I think it definitely worked out the best. I think like in general, like my life is a lot better having had gone to Clemson, having stayed home, stayed near my family, stayed near my now wife.
Right.
She went to school 10 minutes down the road from Clemson.
And then she transferred, right?
Say that again?
Didn't she transfer to Clemson later, or?
So she got her first degree from Southern Wesleyan and then got a second degree at Clemson.
Okay.
So she is currently beating me in degrees.
Oh man! How many more years?
Our dog still has 0, so I'm not at the bottom of the leaderboard yet. But yeah, so I ended up going to Clemson. I stayed in chemical engineering. That was the path that I took. The way Clemson does it is that if you want to declare chemical engineering, you kind of have to do that after your first semester as a freshman. Everybody goes into general engineering. If you want to do chemical engineering, you have to declare that after your like first semester. And there's a specific chemical engineering class for second semester freshmen, while all the other engineers still stay general engineering, and they like take like a MATLAB course or something, and they declare at the end of their freshman year going into their sophomore year.
Why?
Very weird.
Yeah. Do you know why they do that? Why? Why do they have chemical engineering there? Is there like more curriculum to go through?
Their reasoning was that there was no real use in learning MATLAB for the things that we were doing.
Okay. Is that
I
true?
mean, I did get through the first principles things, I guess is not, I mean, obviously like we got a decent education, so it wasn't like necessary for us to learn MATLAB.
Right.
We were able to do things in Excel that had to be done. And so we just took this other class that like they say was more about getting you to think of like a chemical engineer. And overall a good experience. The professor who teaches that class is one of my all-time favorite people. Chris Norfolk is one of the top 5 people at Clemson University.
Wow.
He teaches the first class at Clemson, and that's Chemie 1300 or something like that. He teaches both of the lab classes. He is, we gave him every senior class, like the senior superlative. And we gave Dr. Norfolk the most likely to tear you down on a lab report and then turn around and hype you up to a recruiter to get you a job. So, I mean, he heard the name of the superlative and he just started walking to the front. He's like, that's me.
Yep.
Like one of the most stand-up people you'll ever meet. Absolutely, like will do anything for his students. Like if you put in the work for him, he will like turn it around tenfold. Just to try to get you back. He like his comment, we had like 88 seniors my graduating class. He's like, I want 88 jobs by March or something. He's like, I want you all to be taken care of. And he set up recruiting events, he set up speakers, he did a lot. And so getting to know Dr. Norfolk at the beginning was super awesome too.
Right.
But yeah, I stayed in chemical engineering, and at the time I was just like really convinced I was just going to go directly into industry afterwards. I had an older cousin who graduated from Clemson in chemical engineering. And when he learned I was going into chemical engineering, he's like, hey, Adam, like, I know you didn't ask for my advice, but here it is. If you're going, like, if you're going to do chemie, you need to co-op. Don't do an internship, do a co-op. He said, finding it, I was able to find a job, but it's so much easier if you co-op. You get a year of experience in industry. It is so much better for your career. And I was like, okay.
Can I ask a question real quick?
Yeah.
How long do co-ops usually last? A year, or can you do them for shorter periods of time? And can you explain what a co-op is as well?
Yeah, so I think every school does it a little bit differently.
Okay.
From my experiences talking to like my the people I work with now, but at Clemson, the co-op program, you could start a few different semesters. I guess you could start at any semester. But for chemical engineering, you had to wait until you were at least a second semester sophomore to start the program. And co-oping just like meant, I guess it stood for cooperative education. But we spent 2 years. We basically broke up our junior year into 2 different years, and we would alternate semesters between being in class or being on site at your co-op position. And so it's basically an extended internship.
Okay.
And so I went through the co-op resume and interview days. I interviewed with 5 different companies. I ended up accepting one position in Kingstree, South Carolina at DSM Nutritional Products. I spent the summer after my sophomore year there. I came back to Clemson and did my fall semester of classes. I then went the spring semester back to DSM, back on site. The next summer I spent on campus doing research and taking a class. And then I went back for the fall for my 3rd and final rotation at DSM. And so overall, I got a year's worth of industry experience before I graduated, which was pretty nice. It's a nice program. It's really good. I think part of that, go ahead.
What did you do for them?
So this is kind of,
is it complicated? Bad question.
It's not so complicated. It's just more that there was not as much, at least at my site, that interns could do. I spent a lot of time doing data entry and analytics. I spent a lot of my time working on a continuous improvement initiative for one specific manufacturing line. Our site made omega-3 fish oils.
Oh, okay.
And we made them directly from the algae. So they did like algae fermentation, and then they did the oil extraction processes. And I worked on the aqueous extraction line as they were trying to transition to it's a more productive and honestly more environmentally friendly manufacturing process. And so I spent a lot of time improving that. I spent a lot of time working closely with the operators actually in the plant and tracking and trending data. I didn't really get to do many direct engineering projects. I just like wasn't in their systems, and it was just kind of I was told it would have been a lot to learn like each semester. And so it was just like one of the things where I didn't do as much as I wanted to. But I learned about a lot of what like process engineering was. I worked in the process engineering department. I worked closely with process and project engineers. I learned a lot about like what engineers do on site. And I think I just kind of like left. I actually left after my first, I didn't leave, but like I left my first rotation thinking like, I'm not sure this is fulfilling as I want it to be. So at the end of, that was my first rotation, and it was my summer rotation. Immediately after that, I started in the fall doing research. I was doing computational research actually. And I was not doing well in it, but I was, I really enjoyed the process of research. I was working with Dr. Joseph Scott, who is now at Georgia Tech. Extremely brilliant guy, and he had some projects for me, and he knew I was interested in biology, and so he like helped me develop a project that was somewhat bio-related but also related to his research. And so I spent a lot of time just learning like new tools, like new skills, trying to just like develop this like intellectual space that like wasn't necessarily a part of his lab. And I just enjoyed the process, honestly, of just learning and trying to find something new. And so I did that. I went back for my like spring rotation at DSM. And then while I was there, I ended up emailing another professor saying, hey, can I get some wet lab experience, like working in your lab this summer? I ended up doing that, really enjoyed that, really enjoyed wet lab experience. And then went back for my fall semester at DSM, and the spring the summer previously I'd taken an extra class that I would have taken the following spring. So I only had like half a course load. And so I ended up like working in 3 different research groups at the time.
What?
Yeah. So I like continued with both of those previous professors. And then I like joined a creative inquiry project with a new professor, and joined her lab, worked for her for a semester, and worked on nanoparticle drug delivery.
Oh, wow.
Very different space for me. I only worked for her for a semester. And it was more of a, hey, Dr. Larson, I just don't think I can continue working in 3 different groups. And she was completely understanding. And she's awesome. She's been an advocate for me as well. Even now, I have had great experiences with all the professors that I worked for at Clemson. They were all just really, really great people. And just getting all of that experience, like every area that I worked in, I like really enjoyed. But I think, the area that I worked in the most or the area that I enjoyed the most was working kind of synthetic biology in Dr. Mark Blenner's lab. I had incredible grad student advisors.
Just like me?
Max Hill. I don't think I'm as good as the grad students I had. I'm not gonna lie. Max Hilbert is like, I don't know. I've mentioned several people being in my top 5 people all time, but Max Hilbert is definitely one of them. I worked on his project with him. That was the summer research that I first joined, and that was like my first true wet lab experience. And he was like one of the most patient people. He would like let me do his work, and like he would be like kind of mindful, but he was always patient, and he would just like tell me, and he would just take his time really thoroughly explaining things. And I think that just provided such like a good foundation in research for me that like I wanted to keep doing it. I wanted to keep working with people. And so we were doing like protein engineering work. That was like one aspect of the lab. And then the next summer, I had told Dr. Blenner, I was like, I think I want to go to grad school. And he's like, okay, so we should try to get you towards a publication. And so he helped me get onto another project that was closer to completion. And that project did end up getting published. I think I contributed less to it than I wanted to, but super gracious guy. And again, I just got to learn so much from people about how to do like real research, how to do it thoroughly, and then about the publication process too, which no one tells you about. And it's kind of,
Right.
it was such a cool experience. And so, I mean, after that, I was like, all right, yeah, like I for sure, like I love this research thing. Like I want to keep doing it. I want to, at some point in my life, be able to say like, hey, like I contributed something that didn't just like help the bottom line at a company, that like I created something real that could help people. And so that led me to grad school. I ended up applying to 4 or 5 different schools.
Is that it?
Most of them.
Don't people usually apply to more than that?
Well, there's application fees.
Oh, that's true. Okay.
That's what got me. So.
Okay, that makes sense. Maybe I'm thinking of medical school, because when people tell me they apply for medical school, they're like, yeah, I applied to like 40 schools. And I'm like, what? Okay.
Jeez, yeah. I had a friend who applied to 10. And he's now at Indiana University School of Medicine. I think he's doing great there.
Oh,
but yeah, I applied to 4 or 5, most of them in the Southeast. And then I applied to MIT. I was like, we'll just try it. We'll see. I mean, like, it's probably not going to happen, but like, whatever. And then I like got the call while I was on campus at Clemson. I think I was like doing homework, because MIT's semester schedule is like a month delayed. So I was already like working on my next semester at Clemson, and I was like sitting in our lounge, and like I got a call from a Cambridge area code. I was like, no way, no way. I like walked outside, I like took the call, and I was like kind of shaking. And they told me like, yeah, you've been accepted. I was like, no way, are you sure? And I walked back in to the lounge, and like my friend looks at me and goes, was that MIT? I was like, yes, it was.
I would just start like break into tears. Like,
I tried to call Elena, my wife, and she was in nursing school at the time and doing a clinical, so she didn't even have her phone, and I couldn't talk to her. And I was like, but that was like one of my coolest experiences.
So why did you choose MIT, actually, out of curiosity?
So I went to visit weekend. I was between UT Austin, Texas, and MIT. They were my 2 top schools, both top 10 universities in terms of chemical engineering. I really loved both campuses. I really loved both departments. Kind of the deciding factor that MIT, there seemed to be more opportunities. At Texas, I think there was one professor who I was really interested in working with. I met her on my visit weekend. I really enjoyed talking with her. She seemed like such a great person, in addition to her great research. And then there were a couple of other like professors who I didn't get to know as well. And I just like, I liked the idea of the research, but I wasn't really sure. Here at MIT, it just seemed like it's such a big school with so many opportunities, especially in biology. The biotech community surrounding MIT here in Kendall Square and in Cambridge is like second to none. was doing it was not a one-on-one. They did two prospective grad students to one professor on visit weekends. And I talked to one professor, and I was like, so like what really does like make MIT like number one? Like why do people say that? And he goes, well, if I look out my window, I see Pfizer's right down there, what is it? Genentech or whoever. He just started naming off all these different like biotech companies. Like I could see them from my window. And he said, and those are real, like those are like collaborations. These are people I work with. These are, that's where things happen. I think that's what makes this place special. And I was like, yeah, that's pretty cool. You could just like walk down the road and like you could have lunch with the Pfizer guys or whoever, Biogen, like whoever. And I was like, yeah, no, that sounds really great. And I don't know, there was just something kind of awe-inspiring being on campus. And so
have you heard of all the biotech companies coming up in the Bay Area too? Or is that just me? Like I see it on Twitter all the time and I'm like, geez, it's like the complete polar opposites. Like the East Coast and the West Coast are.
Yeah, that's it. That's exactly right. Right. Like, I mean, biotech, there's two places. There's the Bay Area and then there's Cambridge, Mass.
Yeah.
I'm like, can we go somewhere else? What about the Research Triangle in North Carolina? Like, there's places there. Like, we can spread out people. So yeah.
One type of weather. You either get really warm weather all year round or really cold weather all year round.
Oh my God. Yeah. No kidding.
But back to what you were saying about choosing MIT and ending up at the Galloway lab and everything.
Yeah, and so I ended up choosing MIT. MIT was my last visit, and it ended up being the first weekend of spring break. And so Elena was at Clemson at the time, so we had the same spring break. And she ended up taking the trip. I was like, I'm in Boston. They're paying, for my flights here and there. Anyways, like, let's just stay in Boston for a couple of days, explore it, and see what we think. We might as well. And so we stayed for a few days and toured the city. It was early March or like mid-March, which is still like basically winter in Mass. And where in like South Carolina, it's like 70, 80 degrees. We ended up having a good time. Like we really liked the city, and like we just kind of looked at each other and like, I was like, I like this school. And Elena was like, yeah, I can see it's like a great opportunity. And like, we should try this. We should live in a big city. We should do something different than anything we've ever done before. And so I ended up choosing MIT. And what's crazy is I was like, yeah, there's all these different professors I could work for. And like one of the reasons I came here. And then I ended up joining Katie Galloway's lab, who she was not even listed as a professor when I decided to join.
Wow.
So she had been hired that spring, at the same time that I also like was applying to grad schools. And so by March, my visit weekend, I'm not sure that they definitely hadn't announced that her or Ariel First also joined the department at the same time, but they hadn't announced those hirings yet. I think I was we went in March for my visit weekend. You had to decide by April whether you were going to accept the where you were going to declare for what school am I going to.
Right.
And then I went up in May. It was during finals week, but I was like super fortunate that the way Clemson Kimmy set up your last semester, that there were no finals. And so I didn't have any finals during finals week, thankfully. And so I took like the early part of that week and came up to Boston to look for an apartment, which is an ordeal to say the least.
I heard
ended up finding
the prices are.
I just moved so fast too. I emailed some guy 2 weeks before. I was like, hey, I'm going to be here these weeks. What do you got? What can I see? I want to see something in person. And he said, yeah, honestly, I don't know what I'll have in 2 weeks. Email me 3 days before you come.
That's crazy.
It was probably the most stressful 3 days that I've had.
Yep.
I ended up signing for a place that was like 45 minutes from campus via the T, but they allowed dogs. It was in the budget.
Yep.
It worked. So, but I was here in May, and I like ended up meeting up my grad student buddy from the visit weekend, who was also a Clemson grad. We ended up getting some pizza, talking, and I was like, oh, like, are y'all like hiring? Like, I like kind of looked at like the seminar schedule, and it looked like y'all like were hiring, like, you know, of these people, like, what do you think? And he's like, oh yeah, like we're hiring like a couple people. And he's like, so like, were there any people on that list who stuck out to you? Like, and I was like, well, like I like this person, and then Katie Galloway, like I liked her abstract, they looked cool. He's like, oh yeah, like I think they just announced that to the department the other day or something like that. Like the timeline was perfect. Like it had been like right around the time where they were getting ready to announce it or something. They had let their guys know or whatever. And I was like, that's really cool. And so I went into my first semester at MIT, and I had my first meeting with Katie, and I was like, yeah, I want to join your lab. Your stuff sounds really cool. Professor Galloway is like just a great person to talk to in general too. And we had some really productive meetings to start, and just I spent so much learning about a field I had never heard of before. Stem cell adjacent reprogramming. We can make any type of cell from any type of cell. It's just like, this is incredible. My mind was blown. And she was like, do you want to create like genetic circuits that do that? I was like, yes, that sounds cool, right? And then there was an underlying link to cancer, which is, I think that was like primarily what I was interested in when I came to MIT. I was like, I think I want to do like molecular cancer research, you know, what's going on. There's no better place to do that than the Koch Institute for cancer research. And I was like, that was like one of the reasons that drew me here. And so there was this like cool link between synthetic biology and cancer. And I get to work at that interface, where I'm like studying like cell fate transition, parallel cancer, that like they like are all like are involved the same like proteins and molecules. And it's just such an interesting field.
If I can ask, what is there something in particular in your life that got you interested in cancer research, or was that just kind of something that you've always kind of just been interested in?
Yeah, so I think cancer's touched like a lot of people's lives in general. When I was growing up, there was 2 women who were like my babysitters. I'm like the youngest of 3 boys, and I like, when both my brothers were at school and my parents were teachers, so they were at school, I stayed with two different women who, you know, retired women who were just taking care of children. And my nanny, Nancy Gambrell, she ended up getting diagnosed with breast cancer when she was no longer taking care of me. I like had gotten into elementary school by then, but she ended up, you know, getting diagnosed with breast cancer, and I like sort of remembered, you know, her struggle, and I just I remember it just like really taking a toll on her, and that was just something that I was like, I want to do something about this. And so like between her and my Mimi Doris Hoover, like the 2 incredible women who really helped to shape me when I was growing up. And I don't think I'd be where I am without them. And especially like my nanny, who like, you know, she died from breast cancer. It was just, you know, it was something that deeply impacted me. And I wanted to do something.
Yeah, definitely. Yeah, like obviously, you know, my story, like why I wanted to get into science with my dad and everything. I just think that's something that like, even on your worst days and stuff, you all can always like imagine how much worse it really could be, and like how privileged like you feel to be able to do what you physically can do. Like the fact that we can like get up and go, we can go study, we can go do research and stuff, and some people don't even like have the option to get up or to do anything really. And so that's kind of something that I found in my life, you know, as like an inspiration, I guess.
Right. Yeah.
But going back to your conversation with Dr. Galloway about genetic circuits, when you entered her lab, did you know how to construct genetic circuits already, or is that something that you learned?
So Genetic circuits. I never really built genetic circuits before. I spent a lot of time in the Blender lab learning. I ended up working on, I think, 4 different projects across 4 different semesters, including 3 different summers. And so I like gained this just like very wide like field of like knowledge of like how to do things.
Right.
Like in my first semester, I was doing protein engineering. And so I spent time like learning how to produce and purify enzymes and test them.
Right.
My second summer, I was working on a CRISPR library that someone else had generated, and we were working to characterize it. And so I learned a lot about like Illumina sequencing. I worked with yeast, and so I was doing like yeast mini preps and culture and transformations, and then library generations and like the extra amount of effort that goes into like working with a library rather than like with single plasmids. At one point I started up a bioreactor in the lab because like, hey, my industry experience, they did fermentations on site. Not that I was a part of the fermentations, but
Yeah.
I knew what fermentations were. So I worked on that. And then my final summer, so I worked in the Blender lab one last summer between graduation from Clemson and starting at MIT. And it was another protein engineering project, but it was at the very beginning. So there was still a lot of like molecular cloning going on.
Right.
Trying to create different constructs to test. And so I learned how to clone things, how to build genetic constructs. And that is something that is directly translated to like my work at MIT and building genetic circuits. The mathematical modeling that goes into it, and the actual, control schemes that we try to create within cells, I had no idea. I'd never done. I did take one class at Clemson with Dr. Burt Whistle, who was like a relatively new professor when I was there, on systems biology. And we did, this small scale systems biology networks, dynamic system modeling in his class. And I was like, this is really cool. This is something I'm really interested in. I can learn not only like how cells work, the different control schemes that they have, we can model them with math, we can see like where all this different behavior comes from. And I remember being fascinated by that. And I like started talking with Dr. Galloway about all these things, and it's become part of everything that I do now. And so I just think it's really cool to be able to like make predictions to show mathematically what could happen. And then one of these days, those things are going to like come up in cells and actually work.
Right. Someday.
Someday.
Science is time. Right? Basically.
Yeah.
Like fingers crossed, hoping that things eventually work out.
Yeah. I mean, it's the thing where it's going to work out one time. It's just, do you have all the parameters right going into your experiment? I was setting up an experiment with another undergrad who works with me the other week, and we were testing a genetic circuit that she had built. And it's something that it's temporal control. It was building an incoherent feed-forward loop. And we were looking to see if we could generate a pulse of a fluorescent protein, which we needed to do a time lapse on the microscope of it. We created ways to integrate them into the cells, to integrate the genetic circuit into the cells. And so this was the first time we had tested it. So I had no idea like the parameters set up on the microscope, how long it was going to take to see, activity, if we were going to see activity at all, where all 3 of our all 3 of our constructs that made the genetic circuit, would they all get in together in enough cells for us to see? It was just tough. We tried, a bunch of different parameters. But in the end, we were like just a little bit off. We, had the timing wrong. And it's something that we'd have to set up again. We have more knowledge now, but it's just something that, you iteratively have to figure these things out and figure out what's going to work. We are doing something no one in lab has done. If not, no one else in the world has done, maybe, I don't think that's true for this specific project, but it's just starting something and learning something and just figuring out as you go, what's going to happen next and what you're going to have to do to improve it.
Yeah, definitely. Now, kind of going off of that, I'm kind of curious to hear what your day to day is like as a PhD student. Stress 24/7.
Stress. No, I think there, Professor Galloway talks all the time about things coming in cycles, and I think that's absolutely true. Um, I think everything that I do comes in cycles and waves. And so my specific work is in mammalian synthetic biology, but you can't do anything in mammalian cells without first building your constructs in bacteria.
Right.
Bacteria is the powerhouse of synthetic biology. We make and clone different constructs in E. coli, and so there will be a week where I will spend all week trying to clone different constructs that I need to test. And so a lot of the times I'm spending just time in the bacteria, cloning different things, screening different colonies, trying to find my correct constructs that I had designed previously, probably the week before, and spent like a week designing, a week creating, and then a week testing and like maybe a simpler system before doing a longer term reprogramming experiment with them. And so my weeks, I'd say my day to day just depends on the week that I'm doing. But I get in anywhere between like 8 and 10 in the morning, and staying between 4:30 and like 6:30 for the most part. And and just going and following a project through. And so what that depends on the project. It's a very goal-oriented work. And so I will sit down. I really love it if I can get in before anybody else in the lab. I can't anymore. I've tried. There's a few members in the lab, especially one who she gets there at probably the earliest. But that's not a big deal. But to be able to sit down before people start having meetings or really getting to work or like really moving around, to just sit down and set my goals for the week. Because then if I set goals and things that I want to accomplish and draw out a timeline, then that's really going to determine what goes on. But I think today I actually spent most of my time on my computer. Either I was writing an abstract for a conference, and then I'm trying to learn how to analyze time-lapse microscopy with a tool that another member in our lab created. Awesome, plug for awesome work that Chris Johnston and Conrad Oakes have done to make some really, really cool tools for us to use and create really interesting data. Time resolved data is really, really cool. And so it really depends on the day. I basically spend most of my time learning and designing and creating and just trying to figure out how to do things. And then it's really boring when I'm actually running an experiment for about a week. It's doing things that are somewhat repetitive, from experiment to experiment, but then testing different things at the end of the experiment. And so, there's probably like 7 days of buildup for one experiment. Like, all right, I'm doing all these things. I sure hope that in 7 days it's going to work out and look really cool.
Yeah.
And then be reproduced in the next experiment. But
I remember when I first got into lab, it was pretty terrible because it's so easy to contaminate things, especially when you're first starting out, because you have your pipette and you're just you're going all over the place with it because you don't know, you need to keep it in one spot, otherwise you're gonna touch things, and you find yourself constantly contaminating things, and then you get so mad because you're like, I literally just did this 5 times and now I have to do it again. Oh man, it's stressful. But what I was hearing from that is basically all the different things that science is composed of. And I think from the outside, when you first kind of look at science, it kind of seems like it's more so just completely fact-based. And I mean that in the sense that you don't have to be as creative when you're doing it or designing things, or writing, you were talking about graphic design as a part of science. So did you find that kind of difficult to adapt to when you first got into it? Maybe you weren't expecting all of those other things along with it, or?
it's a good question. So I guess I'm trying to think about like what my like baseline point would be. I was a very poor lab student. And like, the chemistry labs and things, like I was honestly like never really that good at those. And so I really didn't think I would be like that good in lab. Um, when I first got into lab as an undergrad, I think most of my time was spent learning how to do things in lab. And so for the most part, I was like, okay, yeah, like I'm pipetting, like I'm just doing these things, I'm running, I'm taking tests, like whatever. And like my grad student was telling me like what to do. And he's like telling me the important questions that we're asking and things like that. And I think it wasn't until I got to MIT that I really appreciated like science communication and things like that. One of Professor Galloway's like big things is science communication and doing so through illustrations and figures, which I do not have a creative bone in my body. And so that was probably one of the most difficult things to me was Professor Galloway was like, I need you to illustrate what your project is going to be. Like, otherwise you telling people it's going to fall on deaf ears if they don't have a good visual. That was difficult for me. But I think everything else, like I knew at some point, like I would have to write up, what I was doing. But just the constant refinement of your skills, how much time you spend doing these other things that aren't just running experiments. I think that did surprise me. But I guess I was less surprised that I would have to do these things, but more surprised at how much time and effort I would spend learning to write, learning to draw, learning to design things like that.
Okay, yeah, that makes sense. Do you have any pieces of advice for people that might be getting into science or something, like where they should start? Or do you think that's just kind of something that develops as you go?
I really got into science and research as an undergrad. I had planned to go into engineering, and so that was kind of already what I had planned on. If you're starting as like an undergrad who's like not sure if they want to do research or if they want to do industry, I can only encourage you just to reach out to any professor who you think might have interesting research. Chances are they are hiring undergrads or willing to take on someone new who's just interested and enthusiastic. And then understanding when someone tells them that like, I appreciate the opportunity, but I don't think this is for me. I think that there is a ton of opportunity out there that people don't realize. I think it was something that for me, I was super nervous the first time I asked Dr. Blenner if I could join his lab. It was like, hey, you don't know me from Adam, but like, I'm really interested in your research. Like, is there any position? Like, I'd work on any project. Like, I just like, I want to try to learn, some synthetic biology or something. Right. And he was super responsive and like, yeah, like, absolutely. Like, would you be interested in working in this project? Like, or this, like, there's some options. It was super enthusiastic and really encouraging. And it's just not something I expected from a professor, to talk to an undergrad who had really no background. And so, I think that was something that I didn't realize that, professors were also looking to help people and do some of these things. And yeah, there are really good people out there who want to help. For people younger, I think it depends on the situation. In my high school, there was no direct link to research. We had lab classes or labs associated with our classes where we do a few things here and there, but there wasn't great opportunities to really get into a lab or like direct a project or think about a bigger picture of a project. It was more like, here's a lab, here's what it's supposed to tell you, whatever. And so that's something that like I would love to see more of. I've talked to people who did have high school research experiences, but there is open source. I hate to say it, but reading, like there is open access like literature out there, and if you can get into it, and just learning about like all the cool things that people are doing.
Right.
It's there's just so much out there, right? Like there's so much you can learn. There's so much that like just learning about what people do. Like I said, like I got to MIT and I had done research in, you know, a few different fields, and I'd never heard of like reprogramming or things like that. And so, I mean, some of the stuff seems like science fiction, right?
It does.
And so I don't know. I don't know if I have great advice, except for, you know, there are people who like Professor Galloway, like Professor Blenner, Professor Scott, Professor Larson, the people I worked with, who are willing and like able to help get people involved and get people opportunities. And that, you know, all you have to do is ask. And that if you don't know what you want to do, there's literature out there that if you want to get into it, you can certainly learn about interesting things that are going on in the world.
Right. Yeah, definitely. I remember when I was like, as like, I guess it was, I've been working with you guys for like a year and a half. Wait, 2 years, something like a year and a half. Yeah, I don't know for sure.
Was it fall 2020?
Yeah, it must have been. Okay. So,
Yeah.
okay. So I remember though, I just like messaged Dr. Galloway because I was like, oh, this is really cool, like a professor at MIT, and maybe I can work with her. And like I had no idea who I was, so I just emailed her. And then I was talking to her later, and she's like, yeah, I didn't know, because I was like, wow, this is so strange. And then I was like, I remember when I was in high school looking for opportunities, and she's like, might as well, you know, support the next generation. And I'm like, yeah, there are good people. So then when I'm a scientist, I can pass it down in the, like, you know, to more students and stuff. But yeah, that's nice. Like people will give opportunities, because sometimes I feel like scientists are a little bit intimidating to approach. So kind of like what you were saying. But yeah. Um Now I'm kind of curious, you know, you're talking about synthetic biology and everything, and what exactly, like with genetic circuits and everything, but you do a lot of research with P53, right?
Yeah, I do.
What exact like what exactly do you do with that, I guess I should say?
Yeah, so I think my research is kind of two-pronged, and like one part of it is genetic circuits, and the other part is looking at the role of the tumor suppressor protein P53 in reprogramming. And so this is I was talking earlier about the links between reprogramming and cancer, and P53 kind of sits right there at the middle. It's been kind of long accepted that P53 is a barrier to reprogramming, and in some points it is. People have shown extensively that if you knock out or knock down P53 or inhibit it in any way, then you can increase reprogramming efficiencies. And you know, a lot of people, or I guess specifically, I don't know if it's really appreciated, but we kind of parallel this with cancer and the oncogenic transition.
Right.
P53 is the most commonly mutated protein in cancer, and is mutated in 50% of human cancers. And so, you know, if you look at this, it's like, okay, you lose a tumor suppressor, you can reprogram to iPSCs or motor neurons or other things, or you could reprogram to cancer. And so we have kind of that link in mind. But you know, my specific work is kind of looking at how there might be what's the word I'm looking for? Kind of a second role for P53 in reprogramming. We have this really neat system where we have a minimal P53 mutant. So I guess I should take a step back. And so P53, like I said, is the most commonly mutated protein in cancer. And a lot of the times you can get a tumor formed from a cell that had a single mutation of P53, where you have one allele that's still wild type, but the other is has a mutant. And a lot of the times these mutants get referred to as dominant negative mutants, because they kind of take over the cellular phenotypes, and they drive aggressive cancers. In our system, we use a really minimal P53 mutant that completely lacks the DNA binding domain, which is kind of an absurd mutation and is not something that would really exist. or come about naturally, I think. But it allows us to kind of probe different parts of the system. And so it inhibits P53 in one way, but it doesn't degrade P53. Whereas the way people traditionally do that, or the way people traditionally inhibit P53 is using like a knockdown of P53 RNA so that it just doesn't get expressed. And so you have no P53 protein at all. And so our system, it just inhibits one part of the activity of P53 without degrading it. And what we find is that it actually seems to be a better system than when you actually just knock it out completely. And so it's kind of been my job to look at this phenomenon and flesh it out. Like, why could this be? Is this something that like is real? Like, is, P53 has a function as an activator of transcription, and it activates different genes that can lead to cell cycle arrest or even apoptosis and cell death. But if you eliminate those functions, it might still have other functions where it interacts with different proteins at the protein level rather than just activating genes. And how might those interactions be helping not only like, how might those functions actually help reprogramming rather than being a hindrance? And so I'm really, really interested in this dual role of P53, where it can either inhibit reprogramming by causing cell cycle arrest or death, but also promote reprogramming by promoting the cell's genomic stability and keeping it alive.
Right. Can you tell us anything about what you found, or is that like going to come out in a publication and so you have to keep it undercover for now?
I think at this point we are still kind of building better systems to report on P53. It's I don't know. It's the hardest molecule I've worked with, but I haven't worked with that many, right? P53 is normally expressed at so low levels, it's hard to detect. It's very hard to detect in live cells and hard to track it. And that's like one of the things that we're trying to do is create a system where we can live track P53 concentrations and to be able to isolate different subpopulations of cells expressing different P53 behaviors.
Yeah.
And we're close. I have a few systems that they need some tweaking, but I think that we're like nearing a point where we can really test some of these hypotheses and so I should say rigorously test these hypotheses. We have a lot of phenotypes that we observe, both in reprogramming efficiencies and transcription and proliferation states. But to get concrete answers, we really need to be able to isolate these different subpopulations of cells and then track them afterwards, track their reprogramming efficiencies. And so I spent a lot of time thinking about and developing these reporters and creating standards for these reporters and internal standards and thinking about the best way to quantify some of these things. And so I always think I'm 2 weeks away. But I think, we are hopeful that we will have completed the story kind of by the end of the semester here in the next 2 to 3 months.
Wow. Hey, that's better.
Completed's a strong word, right? But
yeah. Is anything ever completed in science? I mean,
yeah, no.
I don't know.
We'll have a story to tell, and then people can work on the extensions of that, right?
Right. So I'm actually kind of curious, because I have heard a lot of like, I guess, professors that have had like labs established for a really long time at universities. How was your experience going into kind of like a new lab, or like you know what I mean?
Yeah. So my undergrad research was primarily in somewhat established labs. I think both professors I worked for were about to get tenure or had just gotten tenure.
Wow.
And then I worked for Dr. Larson, who was a brand new professor. And so I had a little bit of experience in both as an undergrad. I don't think it's hard to really compare being an undergrad researcher and a grad researcher as really a part of the ecosystem of a lab.
Right.
I think I would not change my experience in the world. Being in a new lab has been probably one of the bigger challenges, right? But I think that I've learned a lot more than I kind of could have expected to. When I joined with two other guys, and so the three founders, as we like to joke, right? We joined the lab, and it was us and Katie for a while, along with Myrna, who was the research associate for a little bit before she started her own PhD. And we spent some time, honestly, just figuring out what could go wrong. For the most part, like there were these things, there were techniques that had worked for Professor Galloway when she was at her postdoc, and then she brought them over here, and most of them worked, some of them didn't. And for the things that didn't, it was really unclear as to why it no longer worked. We kind of had to start from the ground up on some protocols, whereas other protocols worked perfectly. There were things where like the cells that we brought over, Professor Galloway had brought over from USC, were not quite great for reprogramming anymore. Like something happened in the liquid nitrogen on the way. I don't know. And so it really kind of hindered the results of our reprogramming. And it took us a while to get our own cells. But once we did, it was like, wow, no, this works great. This works exactly as you described it beforehand. And it's like, okay, just troubleshooting all the little things that come up with starting a lab. It's like, how do we get the protocols that were tried and true in one lab? How do we get those up and running in this lab? And it definitely was difficult, and it didn't help that we had 2 months in lab before the COVID pandemic hit, and we had to leave lab for 4 months. Certainly didn't help, but I think I got to learn a lot about like what starting a lab is like, and then I also got to develop a really good relationship with my advisor, and like how things go. And Professor Galloway is a great mentor, and is really open about how she operates and how like starting up a lab is operating. I think it's currently my goal to also become a professor at a research university and to run a lab. And so like this experience, I think, is just huge in general, just to learn how to start up a lab. And I don't know, I say it a lot, but I really have like learned so many different things, and kind of come up with like ways in which I'm like, I think that we need robustly test things first in this way before we start asking some of these difficult questions. We just, learning a process and how to make things robust and scientific and doing good science. And through trial and error, it's not fun, but it works. And so whenever like a new grad student joins the lab, I'm like, yeah, do this. You want to set up your syringes like this. You want to filter like this, because if you don't, you'll forget and you'll be stuck with one hand trying to open a syringe or doing something like that. I've definitely described my methods and to someone, and I'm like, listen, I do this because I forget and mess up some of these things all the time. So like, if you do it like me, you won't make the same mistakes I did.
Right. So. I remember when I started in the lab, another mess up story. Like, how your pipette tips are in like those containers, like those plastic containers, or I think they're set up the same everywhere, because like, why would they not be? But and you go to press down, and sometimes the container's like not stable because it depends on how the bottom is. And it like flips up. I did that a few times, and I would just be like pressing down, and I would like press down really hard, and then I would come up, I guess, at a really bad angle, because I would just flip the entire container. I was like, oh my gosh, like this is slowing down everybody's work. Like it's not just mine, because everybody uses the pipette tips. So I tried.
I'm in the third year of my PhD, and I did that last week. So.
Okay. So the news is that those mistakes never stop.
Oh yeah, I'd like to blame the person who autoclaves the tips, but that's also me. So, or at least it was.
Fair enough. What year of your PhD are you in right now? 3?
I'm in my third. Yeah. So I started in fall of 2019.
Are you thinking, are you doing 4 years, 5 years? I know some people do 6 or.
So it looks like it's leaning towards 6. I'd prefer 5.5. The pandemic set us back. And biological research is slow, unfortunately. We're at the mercy of ourselves, how fast they want to grow, how fast they want to give us data, and if they want to cooperate that week or not,
Right.
which is not a given. I think 5.5 to 6 years is a realistic timeline.
Okay. Do you know what your plans are for after, like a postdoc? Do you think that you can go straight into a professorship position or?
It's pretty unlikely that I could go straight into a professorship. I think there are some people who have done it. Actually, one of my advisors as an undergrad, I think a lot of people who do that are co-advised. And so they have a pretty unique perspective as opposed to people who come out of grad school with a single advisor. Like people who are co-advised, have 2 different advisors, learn 2 different advisor styles, and can often make that transition a little bit better just because they've got kind of like a broader experience. I think a lot of people do a postdoc anyways. And I think that's my current plan. Just to learn a completely different field. I think that's what a lot of people say, like kind of the best part of being you found a good postdoc when you feel like you're starting all over again. That you are learning a brand new field, you're learning something new. I was actually having this conversation with Professor Galloway last week about, how do you make that transition to a faculty? And I was talking to her, I'm like, yeah, like I have my project, that you gave me, you said, here's this topic, like what's interesting about it. I was like, I can do that. Like, I know how to ask the interesting questions. It's just like when someone doesn't give me a topic, like, how do I do it? And so she just talked about, you take what you learn in grad school, you go learn something completely new as a postdoc, and then you find the synergy between them, and see like how can you create something new at the intersection of 2 different fields. And so my current plan is to postdoc. Where or in what, I have no idea. It just depends on where the next couple years go, and then what opportunities are out there come that time.
That's rough though. I mean, 6 years of like studying something and getting so familiar with it and then going to something else and just being like, I have no idea what I'm doing.
I don't, yes. However, there's something about doing research that does stick with you. We just hired a new postdoc in our lab who is an incredibly gifted immunologist. And he's awesome to talk to and ask really, really good questions, but he has no background in synthetic biology or cloning or doing the reprogramming stuff we do. Right. And so, I've been working with him to catch him up on some things. But I mean, he's a postdoc, he's done research before. And so he's really great at learning, learning fast. And so the learning curve should be, not as steep, right? You know how to do research, you know how to read literature. And you get to bring a really cool new perspective to a different field of research, which, like I said, it's been awesome talking with this guy. Just completely different point of view.
Yeah, definitely. Talking about the time commitment, kind of not necessarily required, because I know they can be flexible depending on like who your advisor is and stuff. But like, how many hours would you say a week that you spend in lab, and then how many hours altogether do you spend just like working on your? I'm scared to ask about it.
I
mean,
I think it's. I can't say specifically like lab, or I can't do like a real breakdown. Like I said, because it just changes week to week. I think overall I spend 50 to 60 hours a week working on my PhD in some sense. Yeah. I'm not the most focused worker, so I could probably do it a little in less time if I were, more focused on the task at hand doing something. In lab, I could probably get away with like physically doing things in half the time. If I'm doing tissue culture and setting up a reprogramming experiment, I think that single experiment is going to take up at most 4 hours a day. most of the time 2 hours or less, just physically pipetting and moving things around and setting up the experiment. The last day of an experiment can take longer, depending on what you're doing. If you're staining, if you're doing all these different things, it can take longer. But so much of what we do, requires you do something and then for 2 hours and then you don't do anything until the next day. Or if you're doing cloning, you set something up and then it runs for 45 minutes. So what else are you going to do? Right. There's certainly times where I can spend 9 a.m. to 5 p.m. just in lab, not even open my computer. And there have been days where I don't even take my computer out of my book bag just because I know what I'm doing that day. I'm going to set up this, and then that's going to run here. And I just kind of set it up staggered so that it all is running. But I think that's not always the case. And so a lot of the times I spend at my computer, like I said, designing constructs and designing circuits and designing different fusion proteins. That takes a lot of time. Designing a lot of cloning vectors takes longer than you think, and a lot of focus. Writing takes time. Learning analysis tools, so much more of my time is spent analyzing the data that I collected than I thought it would be. I honestly, there's sometimes where I think I spend more time analyzing than doing the actual experiment. Yeah. Because there's just so much interesting things there. We like to collect really thorough data sets. And so there's just so much more to do.
Right. So it's analyze, data, cry, experiment, repeat.
Yeah.
I get this from PhD comics because I see those sometimes and I'm just I wonder how Adam would feel about this.
Oh my God. There was this one experiment. Actually, it was a combination of two experiments. I was setting up this P53 reporter and I set it up with a co-transcribed fluorescent reporter. It's supposed to be a blue fluorescent protein. Yeah. Whenever I express the blue protein, the BFP by itself, it's bright. It looks very distinct from a cell that does not have the BFP in it. It's very nice. Whenever I co-express it with my P53 reporter, it's so dim, it's not even higher than the autofluorescence of the cells. And I'm how does this make sense? This does not make, you are still a protein. You're still a fluorescent protein. You're being expressed, not linked to anything. I don't understand. And so I set it up in two different ways to co-express it in two completely different mechanisms, and they both were bad. And it takes 7 days from the beginning of a reprogramming experiment to 4 days post-infection, which is our normal intermediate day that we test things at. And I was super excited for the second one to work because I was it's a T2A. These things work. People do this all the time. It's gonna be fine. And then I get to that day and it's still bad. So I'm why? Just cry,
Did you find out why?
No one knows. It's this protein's being super weird for several other people in lab in completely different contexts.
Are you sure it was there?
Yeah. Oh, so this is the thing though. I created this reporter. It's supposed to be kind of a noise reporter. And so the idea is to quantify the P53 signal relative to the blue signal. Since they're co-expressed, they should be the same within a cell. And so it's kind of a standard within a single cell. Right. And so there is a true relationship, because you can see the P53 signal, or actually a different tagged protein that I had as a control. You can see that clear as day above background. And then you see it and it has a relationship with the blue that's not there when you don't express the blue with it. And I'm so you're there, you're just not bright anymore. And it makes no sense. Nothing makes sense.
Nothing makes sense anymore. You started and you're just my hope is gone. Everything's science-wise.
This is the thing where people synthetic biology in immortalized 293 T cell lines, oh yeah, strong, whatever. And then it's synthetic biology in primary cell lines, and it's oh gosh, we don't know what we're doing. No one's done it, and and no one's robustly done it at least. And it's why are these things? Why are you the way that you are?
Yeah. So. Yeah. But going off of your schedule, I was wanting to ask, like your free time and stuff? Because obviously being a PhD student, as we've gathered, can be stressful. It can also be very rewarding but stressful. So what do you do in your free time? Sleep?
Yeah. So I joined a year and a half ago. I joined a CrossFit gym because I wanted to get exercise, and I like have tried the whole like just get a gym membership and work out on your own thing and failed miserably at that. I have found that I need someone telling me exactly what to do in the gym that day, for how long, for how many reps,
Yeah.
or whatever, or I just won't do it. And so Elena, my wife, found this gym that's a 7-minute walk from my work, seven from my lab. And we joined CrossFit a year and a half ago, and I really enjoyed the community. And we started out going like 2 or 3 times a week, and now we go like 4 or 5 times a week because we really like the community. I've like gotten into the best shape of my life. Like I really have enjoyed it. I hit like a personal like record the other day. Not a record, but like it was kind of a big moment, like a personal milestone.
Oh yeah.
So I hit like a muscle up, which was like something I never thought I could do before.
Wow.
And I was named member of the month at our gym
Oh my
for the month
gosh.
of February. So super proud. I'm the mom. That's been like probably the coolest or one of the most consistent things that we do. My wife and I like to take our dog on, we call them hikes, but like we're just walking in nature, right?
Yeah.
We went out yesterday to Spot Pond, which is like north of Cambridge. It's currently completely frozen over. And we just kind of walked. We actually walked across the frozen pond, walked around nature, just like being outside, walking. We walk our dog all the time. We try to give her as much outside time as possible. She's a mess, and we love her.
Oh, you're in an apartment building right now, right? So there's not like a backyard or something. So you guys take her out. Okay, yeah.
There is a backyard. It's super small. It's not fenced in, and there's very specific rules about like animals in our building. And so we don't take her out there all that much.
Right. You don't want to risk anything. Yeah.
We love where they live, and we don't want them to give them any reason to evict us. So.
Yeah, fair enough. But I was gonna say CrossFit's good because in high school you played like 3 sports or something like that, right? So
2, yeah. I played football and baseball in high school, and so it's nice to be able to kind of lift heavy and do some of the things I was able to do in high school with like a group of people who are like encouraging you to just like keep going. It was definitely like the best part of lifting as a team, and like now like in the CrossFit community too. I try not to like mention too much that I do CrossFit because I don't want to be that guy who's like, let me tell you about it.
that CrossFit guy that's always telling you about how long his workouts are and everything.
Exactly. I really try not to be that guy. And so I'm just like, yeah, like I work out. I just don't tell people what I do. But I can't like say enough about just kind of like working out with a group of people and like having a coach tell you what to do. It's been in terms of physical health, it's the best I've ever been. I went to the doctor the other day, and like my resting heart rate was like 59, which is like I've never seen that before in myself.
That's really low. That's good.
It is like Elena's a runner, and so hers has always been like 50s. Sometimes like one time she measured it in the 40s, and I was like, "Are you alive?" Like, but like when I like got in there 60, I was like, I called her. I was like, "Yo, you got to hear what they said." And like the nurse asked me if I like worked out. I was like, yeah, I do. Like, I don't know. I was just super excited. Like, cause
then you go into your school about, I do CrossFit. I do this.
I didn't say it, but I was, it was just really nice to like actually like, have gotten to a level of like physical health that I like haven't had in a really long time.
Right, definitely. And talking about sports, that's another thing that you keep up with, like watching sports and stuff, watching Notre Dame dominate all the time, right?
Yeah, not Notre Dame, no, for sure. I'm actually wearing my my
Oh
Braves
my.
World Series championship shirt.
Okay.
Huge Atlanta Braves fan.
Hmm.
Huge Clemson Tiger football fan, as they have
I'm gonna have to edit that out.
beat
We
the
can't talk
tar out of
about it.
Notre Dame like 3 out of the past 4 times they've played
Oh
them.
man, we're gonna have to go to the next topic right now.
Any meaningful game that Clemson and Notre Dame have played in, Clemson has just smashed the tar out of them, and it's been fantastic.
Well,
They play again next year.
Clemson's not doing amazing now, though. So
We still got the 10 wins this year. You know, that's the thing, right? Like,
that's true.
it's a down year from Clemson, and we still won 10 games.
You guys take what you can get.
Yeah.
What you can get is still better than what most teams can get. So it doesn't really matter anyways.
We'll see. Clemson comes to Notre Dame this year.
And you won't want to watch that game because you're going to be very disappointed. I hate to break it to you, but you're
I'm probably going to be very busy. We'll see, but I don't know if I'll be disappointed.
You might not, I'm gonna have to like message you updates like this just happened. You're like in lab, like celebrating everybody. What is Autumn doing over there?
I'll have like the iPad just sitting up with the game as I'm like cloning and just like taking a colony, like go 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 go.
They're like, are you talking to yourselves? Like, what's happening right now?
Yes, also encouraging them.
Yes.
Please grow.
Yeah, but anyways though, to close out, I just kind of wanted to ask you about any words of encouragement you might have for people, like just like kind of last words of encouragement to kind of summarize what you've already said, for people that are looking to get into engineering, STEM, just in any capacity.
Yeah. I don't know how encouraging it is, but I mean, just the science and engineering and research are hard. They are not easy. They are things that have taken people years and years and years to refine. And like, especially doing research, you're doing something that no one's ever done before. So like when people begin to struggle, when you start to think that like, this isn't for me, like I just can't do it, I think it's just super important to remember that like pretty much everyone has felt that way. I know like I went from Clemson where I was, making A's, doing well. And then I got to MIT my first semester and I felt like I was drowning. It was just one of the things I was like, this is so difficult. This is so out of my league. Like, did they send the acceptance letter to the right person? But I think that it's just what do they say? Like anything that's worth doing is going to be hard, right? It's not, if it were easy, someone would have already done it. And so science and engineering and research are things that are hard, but they're worth doing, and that it doesn't take some innate special ability. It just takes someone who's willing to work hard, to work past the failures, to learn from their failures, to just keep going. Someone to think a little bit differently about a problem that someone else hasn't. And so if you ever feel like you don't belong, or like if you're like not like up to it, everyone else has felt like that too, and that it's normal to feel that way, but you can break through.
Yeah, definitely. Yeah, like what you were saying, I guess, because I think like even as like an undergrad, like your course load isn't, or like workload, I guess I should say, overall is not as heavy as like graduate students. But I think it's easy to feel like discouraged in different ways at each stage that you're in. But the thing is that everybody else has felt that way at some point. They just don't feel it now. Like the people that you look up to just don't feel it now because they've made it past that and have been like gotten like more experience doing other things. And so like in order to get to that, it's like requires a lot of grit and perseverance, I guess I would say.
Absolutely.
But yeah, thank you for joining me. This was a lot of fun. So yeah, thanks.
Thanks for having me.
Thank you for watching. Don't forget to like and subscribe for future videos. Follow our Facebook, Instagram, and Twitter for future updates.